Welcome, and thank you for joining me today. I'm Lindsay Hansen. I am a fish ecologist with the U.S. Geological Survey's Grand Canyon Monitoring and Research Center. And today I would like to share with you a novel tool that may help improve predictions of fish growth in resource-limited river systems. In the existing literature, there have been an assortment of different types of data that have been used as proxies for autochthonous food availability in river systems. So proxies for the amount of algae and aquatic plants that are growing within the river itself. These measurements of ecosystem productivity include the normalized difference vegetation index, or NDVI, satellite-based estimates of chlorophyll A concentration, and daily variation in dissolved oxygen. So while these tools are useful in some systems, they may not be especially useful in rivers and streams, especially in those that are resource limited. So the research I am sharing with you today occurs in the Grand Canyon portion of the Colorado River, which is characterized by low standing stocks of producer biomass, and the food available in this system is not indicative of the production and consumption rates within the river. Increasingly available in the past around 5 to 10 years, though, is high frequency gross primary productivity data. This type of data is modeled based on daily dissolved oxygen, water temperature, discharge, and canyon shading measurements. So these models allow us to estimate the rate of food production in rivers where food webs are driven by autochthonous production. GPP, as I'll call gross primary productivity throughout this presentation, is a more appropriate measurement of primary production in the Grand Canyon and in systems like it, because the system is primarily autochthonous and because the bubbles formed in the rapids of this reach of the river lead to an oversaturation of oxygen, which makes it kind of impossible to estimate daily values of ecosystem respiration from dissolved oxygen alone. So we do need all of those other things to add into the model, all those other variables to get an accurate measurement of that kind of autochthonous food. GPP has been used as a tool for predicting fish production, but that data from these new models has not, at least to our understanding, been used to predict freshwater fish growth. And we're really excited to have been able to test these data in our models. So our story begins with the Colorado River. Before major development, this river was extremely dynamic. It had flows that ranged from huge floods to just slight trickles, and temperatures that ranged from freezing to 35 degrees Celsius, so a really big range. Beginning in the 1900s, the Colorado River system has been allocated for irrigation and now has many major dams that fragment it. And it now has the unfortunate distinction of being the most regulated river system in the United States. Our study focuses on the Grand Canyon portion of the Colorado River, which is separated from upriver by Glen Canyon Dam and stretches for 300 kilometers downstream until it hits Lake Mead. In this river is an endemic primary consumer fish species known as the flannelmouth sucker, and I'll probably refer to them as flannies throughout this presentation. They are known to feed on invertebrate prey and algae. They're quite long-lived. They can live to be about 30 years old. And they're abundant within this river. So there's a significant quantity of growth data available for this population, and that's why we've used them as the study species for our research. Glen Canyon Dam blocks sediment and warm water from upstream from flowing into the Grand Canyon and has made the river water cold, clear, and less allochthonous than it was pre-dam. So in accordance with the serial discontinuity concept, the river gets more dynamic, more turbid, warmer with increasing distance downstream from the dam. So as tributaries bring in different water from different side canyons, it mixes with the water from the dam, and then it also has more time in the sunlight to get warmed up. So the kind of mixing of conditions creates kind of a continuum of habitat. Because these environmental conditions characterize the Grand Canyon, and because temperature and food or resource availability are both known to influence fish growth, we used water temperature, gross primary productivity, and turbidity as a proxy for the habitat conditions that would influence growth. So we use GPP as a proxy for autochthonous food inputs, and we use turbidity as a proxy for allochthonous food inputs to model flannelmouth suckerfish growth within the Grand Canyon. So Grand Canyon is a really large system, and the Flanny research that has occurred there is a conglomeration of efforts by many organizations, including the USGS's Grand Canyon Monitoring and Research Center, U.S. Fish and Wildlife Service, the Arizona Game and Fish Department, and the National Park Service. So technicians from these organizations used hoop nets and nighttime boat electrofishing to catch flannies. Each flanny was either scanned for or administered a new pit tag, and their length was recorded. And we stratified flanny growth by monthly intervals over a 6-year period from 2012 to 2018. We split our study area into 4 reaches to reflect the changing environmental conditions created by major tributary inputs. So this is a map of the Grand Canyon such that water flows from the right to the left. Glen Canyon Dam is up here on the top right corner. And some of the most major tributary inputs across the 370-kilometer canyon are here. So each tributary brings in a unique contribution of water into the Colorado River. It introduces runoff from different rock types. There's different amounts of discharge and flooding frequency, and there's a unique nutrient and allochthonous food input from each of those side canyons. And it was for this reason of like mixing waters that creates different habitat conditions from upstream that we separated the river into these 4 sections specifically. Our environmental data was obtained using stream gauges and YSIs at the confluences of each of those major tributaries. We obtained monthly mean water temperature and turbidity data from USGS stream gauges, and our GPP data was modeled using dissolved oxygen data obtained from YSI probes in the same locations. We built, fit, and selected state space Bayesian growth models that used turbidity, temperature, and GPP to predict flannelmouth sucker growth and length. And we found that in our system, flannies grow more quickly in warmer conditions with a higher proportion of benthic diatoms and algae, so with higher GPP, but that their growth slows down in months that have a lot of highly turbid days. So growth increased with increasing temperature and GPP, but it decreased with increasing turbidity. So taking a look at this plot, we can see that mean growth is in this bold black line that's kind of in the center. And this is the mean growth rate for flannelmouth suckers if all of those environmental conditions are at their means. So we can see that a small young fish will grow around 7 millimeters per month. But as we get to those kind of larger fish, which are on the right-hand side of this graph, that growth slows down, which makes sense. If you're a younger, smaller fish, you're going to grow faster. And then as you become an adult, you get bigger, your growth is going to slow down. From here, we can see how growth will differ under more variable habitat conditions. So growth in highly turbid waters is this solid purple line. And then growth in low turbidity conditions is the dotted purple line. And so we can see that when temperature and GPP are at their mean, if we have a really high turbidity season, that growth is going to be really low. But then if we have a season with lower turbidity, that growth will actually increase. So we can see this kind of opposite trends in both GPP and temperature. So GPP is represented by this green line, with a high GPP is the solid line and a low GPP is this dotted line. And we can see that when GPP is higher, growth increases. And we can see the same trend for temperature, except for the trend for temperature is a little bit more pronounced because temperature is kind of the driving factor of growth in this system. So this finding that temperature has the most influence on growth is consistent with the growth literature for ectotherms, and it supports growth models for other native fish in the Grand Canyon. So even though temperature is that biggest driver, we did find that allochthonous and autochthonous food resources can each explain about 50% as much of the spatiotemporal variation of the growth of an average size flanny as temperature does. So to the best of our knowledge, this study is the first to test and show that these novel high frequency GPP data available as a proxy for stream metabolism and resource availability impacts freshwater fish growth. So in this figure, we can kind of trace the trends in growth in a year as they follow the mean conditions for both temperature and GPP. So here, the blue line represents the mean monthly growth in millimeters of a flannelmouth sucker. Each of these panels represents a different reach. So reach 1 is the closest to the dam, reach 4 is the farthest away from the dam, and the day of the year is on the bottom on the x-axis here. And so if we take a look at more of these graphs, we can see that just like in the last figure, the green represents GPP, the purple represents turbidity, and the red represents temperature. So if we look here in this reach 1, we can kind of see this bimodal pattern of growth. And that kind of follows the patterns of temperature and GPP in the system. So in reach 1, we see this big spike in GPP kind of in the spring, and that is followed by this big kind of hump of growth during that time. And then as we get to kind of later in the season, we see that temperature increases. And so we also see kind of a boost in growth during that temperature boost as well. If we move over kind of farther down away from the dam, we can kind of see how here temperature is really, really high in reach 4 because we do get the influence of the solar insolation warming up the water and some tributary inputs. And so growth is highest, is high in these bottom reaches where temperature is really high. We do think that the reason why growth is a little bit lower here in reach 4, even though temperature is a lot higher, is a mixture of this GPP being a little bit lower and our turbidity kind of having this, this spike. We were surprised to find that in months with more days with high turbidity, flanny growth rates decreased. And so although we had used turbidity as a proxy for allochthonous food resources, seasonal monsoons and snowmelt create intense turbidity conditions within the Grand Canyon. Flannies are sucker fish. They have terminal mouths. They consume food that exists along the substrate of the river. And so these high turbidity conditions may inhibit the flannies from successfully locating and consuming food because these flooding conditions stir up the food resources within the river. This is likely why we see in that reach 3, even though we have that like high temperature and high GPP, that growth isn't extraordinarily high because there is that high turbidity within that reach. So within ecosystems like the one that we are working in, where fish growth is linked to stream metabolism, the variation in daily stream metabolism in analyses may allow managers to identify conditions that promote increased fish growth and may enable us to have dam management practices that are better for native fish growth. So as an example, let's consider a separate study by my co-author, Bridget Deemer, in which she estimates the change in GPP that will occur under different dam management practices. So currently, water is released from the hydroelectric dam in a predictable and tidal fashion, meaning that more water is released during the day when electricity demand is high, and less water is released at night when the demand is lower. During the 2020 to 2021 year, dam operators partnered with science to have some experimental water releases where the weekday releases followed a normal pattern, but on the weekends, there was a low and steady flow. And the idea with this was to allow aquatic invertebrates to lay their eggs more successfully. This is called a bug flow because it was for the invertebrates. And even though it was for the bugs, it had the unintended result of also increasing GPP by 38% canyon-wide during May and June. So we can then expand that finding to our study of flannelmouth sucker growth. If the mean growth under the environmental conditions made with that predictable normal flow release from the dam, growth is around 10 millimeters per month on average for small fish and decreases as the fish get bigger up until they're about 500 millimeters long. If we were to update our model to reflect that 38% increase in GPP under these experimental flow conditions, we see that this 38% GPP increase would correspond to an increase in flanny growth of 1.6 millimeters per month, or approximately the same effect as warming the river by 1.1 degrees Celsius. So this shows that linking GPP to fish growth is useful for making inferences about the drivers of GPP on shorter timescales and can help us manage the dam in a different way. We encourage considering using high frequency gross primary productivity data as a predictor within growth models, especially for low trophic level consumers and in river systems with low standing stocks of primary producers and high dependence on autochthony. Linking stream metabolism and water temperature to fish growth may be beneficial to predicting how fish populations will respond to increasing water temperatures and changes to the frequency of drought and flooding under global climate change. The data used in the study was made possible by joint efforts from USGS, GCMRC, US Fish and Wildlife, Arizona Game and Fish, the National Park Service, and the Bureau of Reclamation. We're thankful to these agencies for the countless hours put in by field technicians, by boatmen and scientists in collecting these data. And here's also a few other names of specific people who were helpful on this research. And finally, I would like to thank you all for tuning in to this talk today. Please, if you have any questions, feel free to email me or put them below. And thank you for watching.